Hello, in this video we will discuss about the protein absorption and metabolism. So first of all, we will understand about the protein absorption in the digestive system. So during the food is made up of protein. For example, chicken, egg, fish and milk. These are all content of foods contain a protein. So what is protein? Protein is the sequence of the amino acid in the form of peptide bond, which that is in the form of polypeptide bond. So here I am drawing the structure of the polypeptide. For example, these polypeptides contain a CN bond. That CN bond is known as the peptide bond. Each amino acid contains a CN bond to bind with it in the polypeptide. So here is the N terminus and here is the carboxy terminus, which that is important for the digestion because the specific enzyme used for the N terminus dissociation of the amino acid and carboxy terminus dissociation. So the CN bond is the double bond characters in amide is the peptide bonds. And here you can see this is the amino acid which can be delocalized here the bond, and in this way here is the amino group which can be dissociate and negative and positive charge create. So in this way donation of the proton and accept of the proton. So in this way the amino acid in the form of acid, so that is why it is known as acid, because the lone pair are present which can donate the proton. So here you can see the amino acid, two amino acid bind together, and during the condensation reaction the water will be removed, so to form a peptide bond. This peptide bond is the character is the CN bond. Here you can see. So here we will discuss about the digestion of protein during the eating time. So the mastication will be occur first. During the mastication, the lubrication of the food will be occur, and the intrinsic factor, which that is salivary gland, parotid gland, will create the intrinsic factor embed into the protein. And in this way, here is the ingestion will be occur. A peristaltic movement will allow to move in the food into the stomach. So when go into the stomach, so here is the intrinsic factor for vitamin B12 binding through a parietal cell. So the parietal cell is responsible for the production of the hydrochloric acid also. So in this way the pepsinogen will be pepsinogen is in the form of inactive produced from the chief cell of the stomach. So the stomach contain a gastric pict. I am not drawing here. So here is the pepsin. This pepsin is converted due to the pepsinogen inactivation to activate through a hydrochloric acid to convert pepsin. This pepsin is the digesting enzyme which digests the semi-digestion of the bolus, and this bolus will be digested partially, and this is partial digestion will be occur, and in this way this is the type of polypeptide will be formed, but the oligopeptide also. So in this way, after this, the digestion, the semi-digestion or partial digestion will be occur, and in this way the chyme produced. This chyme will move into the duodenum, and whenever it will be moved into the duodenum, the pancreatic enzyme will be produced exocrinally. So the pancreatic enzyme is known as the trypsin and chymotrypsin. We will discuss also. So that is the basically chymotrypsinogen is in the form of inactive to active. So trypsin is used for it. So in this way here is the complete digestion will be occurring in the duodenum first of all. So here is the endopeptides enzyme. In this way the endopeptide enzyme is used for the not N-terminal and carboxy terminal separation, but the digestion of the protein between the polypeptide. So in this way the trypsinogen will convert into the trypsin in the presence of the enteropeptidase enzyme. This enteropeptidase enzyme is basically digest the inside of the polypeptide. So in this way the arginine and lysine will be separate. So on the other hand, these trypsin also stimulate the chymotrypsin again and again, and whenever the protein will be more. So the chymotrypsinogen will be converted into the chymotrypsin in the presence of trypsin. So this will use for the tryptophan, phenylalanine and tyrosine and leucine separation from the polypeptide. So on the other hand, important is the proelastase enzyme. This is the inactive and to become elastase in the presence of the trypsin, and this is important for the separation of alanine amino acid, glycine amino acid and serine amino acid. So this is a pro-carboxypeptidase enzyme is another type of the enzyme which that is the pro, and after the trypsin will active to carboxypeptidase formation, and this is the basic amino acid separation. And in this way important thing is that the trypsin is important enzyme which that is activate the other enzyme for digestion of protein. So here is the protein which that is present in the duodenum. So the exopeptidase enzyme also present in the wall of the plasma membrane of the enterocytes. Enterocyte is also known as the brush border cells contain a brush border enzyme. So inside is a lumen which contain a protein. So I hope you make sense about the excretion of the pancreas and digestion of the protein. But here we will discuss about the amino peptidase, mainly dipeptidase and tripeptidase. So these dipeptidase and tripeptidase, which that is hydrolyze the peptide bond through a dipeptide and tripeptide. So here is oligopeptidase also present in the plasma membrane in the brush border cell on the upper side. So it can be converted into the tripeptide, and tripeptidase enzyme used for the dipeptide conversion, and dipeptide in the presence of dipeptidase which that is similar. So here is the sodium co-transporter channel which that is allowed to move inside one amino acid or two amino acid. So one more important thing is that the hydrogen ion, the proton, is also required for the movement influx of the amino acid. So here you can see. So the dipeptidase and tripeptidase enzyme is also present in the inside of the enterocyte, which that is further hydrolyze this bond, peptide bond. So here is the inside is the down is you can see that is the channel which that is the amino transferase channel, basically amino acid transferase channel, which that is transfer, basically it is the transporter, and it will be transported into the blood circulatory system. So in this way the absorption will be occur. So the complete absorption occur in the duodenum. So after the absorption of the amino acid into the blood circulatory system from the duodenum, from the enterocyte, and that will be occur. So here is you can see the blood vessel, and here is the between the blood vessel and the enterocyte is the interstitial space. This interstitial space is used for the movement of the amino acid into the blood circulatory system. So this is important and used for the absorption. So how it's possible? Because the amino acids are less in the circulatory system will allow to diffusion inside into the blood circulatory system. So here is the portal vein which that is used for the amino acid to the transport into the liver, into the hepatocyte. And what will be happen into the hepatocyte we will discuss here. So this is the liver contain a hepatocyte. So I am zooming out the hepatocyte cell. For example, here is the hepatocyte. So first of all, I want to say that is a high protein meal. During the high protein diet, the gut and the liver utilize most of the absorbed amino acid. So in this way, the more absorption of the amino acid will be more utilization as compared to the sugar. So the amino acid as a energy, also gut uses branch chain amino acid, and liver take through portal vein 60 to 70% these amino acid converted into glucose. And this process is known as the gluconeogenesis because the glucose is produced from the protein. So here I am drawing the graph for understanding as well. For example, during the glucose will not intake, for example, the high amount of the protein will maintain the glucose level in the blood. Why? Because the glucose is not coming in the from the diet, so the glucose level will be equal and the high protein meal during. So it means the amino acid also produce the glucose through a gluconeogenesis. So here is the insulin, for example, which that is drastically increased for the absorption of the amino acid and ultimately will be balanced into the 10 to 20 micro milliliter. So on the other hand, the glucagon is basically also increased during the but drastically decreased due to the increasing of the glucose. So that is important things because the glucose will be also move into the cell, so that is reverse mechanism. So glucagon will be reuptake into the blood circulatory system. So here is the amino acid in the hepatocyte we will discuss, which that is converted into the protein in the form of high density lipoprotein, and it is the transporter into the different organ. While on the other hand, the synthesis of other nitrogen containing compound will be occur. This nitrogen containing compound we will discuss also, and here is the deamination will be occur is if the amino acid will be excess. So the amine group will be separate, and in this way that this amine group is used for another mechanism, or you go into the urea cycle. So here is the synthesis of other nitrogen containing compound. It can be GABA, glutathione, heme, and nicotinamide, adenosine diphosphate, serotonin, melatonin, norepinephrine, epinephrine, and histamine, and for sphingosine. This is the major example, for example, and this is the example of the synthesis of other nitrogen containing compound. While the deamination will be separate the amine group, and this amine group, and here is the transamination will be occur. This transamination means the alpha keto acid will be converted again into the amino acid, while the amino acid is also converted into the alpha keto acid. This is the carbon skeleton. This alpha keto acid we will discuss also. So first of all, here is the alpha ketoglutarate. This alpha ketoglutarate is produced and converted into the glutamic acid and glutamine. So here the ketone body is also produced from the alpha keto acid, and also acetyl CoA will be converted, and this used for the tricarboxylic acid cycle, and while the glucose is through a gluconeogenesis. So in this way, here is the glutamic acid, which that is converted into the glutamine, and this is here you can see the carbon dioxide will be combined to the with the amine group to produce the carbamoyl phosphate, and in this way the carbamoyl phosphate is used for the urea cycle. Either this amine group is used for the excretion, we will discuss also. So the uric acid and creatine is also produced from the nitrogen containing compound. So here is the carbamoyl phosphate, which that is convert the citrulline with the aspartate and arginosuccinate to produce the arginine, and this arginine will separate the urea, and again ornithine will be formed, and in this way the urea will be separate, and this is the basic creatine, amine, and uric acid. Urea, that is a waste product if the not useful. So in the blood is the toxin can create from the excess of the urea and creatine. So in this way it must be excrete. So here the gluconeogenesis will produce glucose, and tricarboxylic acid cycle produce the carbon dioxide and ATP. This ATP used for the different purpose for energy. So here the excretion through a nephron in the kidney, which that is allowed to move the waste into the circulatory system to move into the kidney, and in this way the nephron will be excrete. So here is the next nephron contain proximal convoluted tubule, loop of Henle, and distal convoluted tubule and collecting duct, and the urine will be formed. So this is the Bowman capsule and glomerulus. So I hope you make sense about these things, that is the protein absorption and metabolism. That was the overview. In the next video we will discuss about the deamination and transamination. So thanks for watching. Please make sure to subscribe, like,